Hello, good afternoon, everyone. Welcome to this week's Biome Seminar. Today, we are excited to learn about the effects of climate change on biotic interactions in temperate forests from Dr. Benjamin Lee. Over the last 15 years, the Yale School of Environment has held this weekly Biome Seminar series. BIOME stands for Bridging Issues and Optimizing Methods in Environmental Studies. BIOMES is the school's flagship forum series for bringing cutting-edge research and impactful work to the community. I'm honored to introduce Dr. Benjamin Lee. Dr. Lee is Assistant Professor of Forest Ecology at the Department of Biological Sciences at East Tennessee State University. Dr. Lee is a climate change ecologist primarily interested in how forests will respond to ongoing climate change. He is especially interested in how shifts in phenology alter how plants interact with resource availability and with other organisms. Other research interests of his include species invasions, climate change effects on biodiversity and community composition, and patterns of carbon storage and allocation in trees and forests. Please help me welcome Dr. Benjamin Lee to the stage.
Thank you. All right, hello everyone. Thank you for being here. This is really exciting for me to be here. I did my PhD in an environmental sciences program at the University of Michigan, and so this feels a little bit more like home and the where a lot of the ideas that I'm going to be talking about today were generated, as opposed to like a largely organismal program that I find myself teaching in now. So as mentioned, I am going to be spending a lot of time today talking about climate change and how it affects plants in particular and how they interact with other aspects of the environment. To give you a little bit of perspective for where some of my ideas came from, these are the forests. This is what the forests look like from where I'm from, which is the Pacific Northwest. So very dark, very wet, very constant throughout the year. I mean, there's variation in the water availability, but especially around some of the old growth forests, for example, out on the Olympic Peninsula, the environmental conditions that you find yourself in if you're an understory plant or in the forest understory in general tend to be pretty constant and predictable, especially year to year. And so when I moved out to Michigan to start my PhD, one of the main things that stuck out to me when I moved there was the difference in the seasonality and just how varied forest systems are when you have a primarily deciduous overstory and what that looks like. So for example, even though, as you can see in this forest, it tends to be pretty dark, light tends to be pretty low throughout the year because you have a lot of evergreen trees in the overstory. There might be a couple of deciduous trees here and there. If you are instead in the forest out here, for example, or in Michigan, or even down in Tennessee, especially this time of year, and you were to go and look up, this is what you would see instead. A lot of opening in the canopy. You still see that the sun is being blocked by branches and by trunks as we move, or as it passes through this canopy environment. But the forest floor is a lot brighter. There's a lot more resource availability, especially when it comes to light availability. And over the next couple of months, as the canopy trees, their leaf buds will burst, the leaves will expand, they will start intercepting that light before it reaches the forest floor, which we tend to term as canopy closing. And so as the canopy closes, what you will see instead is something that looks a lot like this. And I don't put these pictures up because I don't think any of you have never been to the forest and looked up at the trees before, but just to really illustrate that there are some patches, like maybe here or here, where the light is penetrating the understory, and we call these canopy gaps. But those gaps tend to be very small and ephemeral in nature. They may only last, if you're a tree seedling or something growing on the forest floor, you may only have access to direct light for 5 minutes a day as that gap sort of passes over you with the movement of the sun. And the difference in the amount of photosynthetically active radiation, so the light that is available to be used for photosynthesis by understory plants, it decreases 3 to 4 orders of magnitudes between the beginning of spring and the middle of summer. So in units of photosynthetic active radiation, it's about 1,200 to 1,500 ppfd in early spring, and it decreases to 5 or 10 during the middle of the growing season. So just this huge contrast and this huge swing in resource availability that's associated with seasonality. And so I became really fascinated with how this variation in resource availability affects the growth strategies and the ways that these plants interact with their environment and with other organisms. And as this resource changes over throughout the year, we see a lot of organisms that really do, at least in the Northern Hemisphere, that really do key in on this pulse of resource availability early in spring. One of the things that I'll be talking about a lot today are spring wildflowers or spring ephemeral wildflowers, like the trillium that you see here, that will emerge up to several weeks before the canopy closes and try to make use of that light availability. They interact with other flowers like trout lilies, like Claytonia, and Dutchman's breeches, which is one of my favorites. Spring ephemerals out here on the East Coast. And so not only are all of these plants trying to do the same thing, which is make use of this high resource availability, but because of that, they're competing for pollinators, they're competing for soil nutrients, they're competing for other resources that may otherwise be limiting. And so when they are active and where they interact and co-occur really shapes a lot of the ecological processes that determine things like their demography. In addition to understory wildflowers, we also have tree seedlings have a very similar strategy. They emerge up to a couple weeks before the canopy closes. We have pollinators that really make use of the highlight availability to be able to navigate the forest understory. We also have other organisms like birds, especially migrating birds moving through these systems, eating the pollinators, potentially helping pollinate the plants as well. Other organisms like ants and deer. All of these organisms really cue off of not only the resource availability, but also the presence of plants as a resource themselves. And so all of this tends to fit together and really form the community structures and the food webs and all of these things that we're interested in as ecologists. And a lot of it is cued primarily by environmental conditions and this swing in resource availability. And so one of the main things that I'm interested in my research is not only the timing of this activity and the relative change in when these plants and when these other organisms are active at a given year at a given location, but also how this shapes their biotic interactions. How does the timing of activity, the duration of overlap in these activity periods, how does that actually affect some of the biotic interactions and some of these higher order interactions that we tend to care a lot about and that we know are tied to ecosystem services and other important aspects of the environment. And so, for example, on the previous slide, we have all sorts of different biotic interactions that are sort of implicitly involved in creating the network. So we have trophic interactions where, for example, deer become active in spring because they like to eat a lot of the understory plants that are coming up early in spring. We have dispersal or seed predation. Pollination is a really big one. Competition with other plants, as well as access to resource availability. And what I'm going to be talking about a lot today, at least for the first half, 2/3 of my talk today, is really cueing in on this resource availability component and this access to spring light. Because a lot of these plants, although the spring ephemerals may be only active for a month or so at the beginning of the growing season during this highlight availability period, organisms like these tree seedlings will maintain activity throughout the growing season. And so how do they cope with the fact that eventually the trees above them are going to leaf out and block that light? What are their strategies to be able to deal with this? How much of their annual carbon budget is really determined by that access to highlight at the beginning of the growing season. And as mentioned, these interactions are really important because they drive a lot of community processes and properties that we're interested in. So things like competitive exclusion, facilitation, that determine where species exist throughout the landscape, how fundamental niches compare to their realized niche, for example, That's determined by these biotic interactions, community composition and structure. This scales up to ecosystem services and function, and even more derived traits like the resilience of a forest to climate change. So all of these are determined in some part by the biotic interactions and the quality of interactions and the extent of those interactions through time. And so when we think about what determines species interactions and what shapes these community dynamics, there are two main things that I tend to think about, at least. The first being that organisms that interact have to be present in the same space. So this is largely determined by environmental filtering, for example. So the environmental conditions have to be suitable to the biology of that species. It's determined a little bit by evolutionary history and dispersal limitations. And so we can, for example, compare the ranges here of two spring wildflowers, yellow trout lily, Erythronium americanum, and jack-in-the-pulpit, Erythema triphyllum. And you can see that here in the bright green, that's where they are common. The dark green is where they're present, but not super common. And then the gold color out west is where they are not present. So both of these are distributed throughout the eastern United States. Both of them co-occur across a wide swath of the Northeast United States. And so they're going to be interacting, at least theoretically, because they occupy the same space. So they may be competing for a lot of the same resources, like nutrient availability, for example. The second criterion, and this is what I'm going to be focusing on a lot today, is that they also need to be present at the same time. So it's not just enough that they are growing in the same woods. They could be growing immediately next to each other. But are they active at the same time of year? Are they competing for resources at the same time? Or is there some sort of temporal partitioning happening where one of these species may be making use of these resources early in the year and the other later in the year? And for example, Erythronium again here, this is data that I pulled off of iNaturalist, which is a community science phenology monitoring platform. And they produce these nice graphics here sort of automatically. And so Erythronium here shown on the left is mostly active across its range from between March and May, so disappearing by the beginning of June. And you can see that the same period for jack-in-the-pulpit here is also the peak of its activity. So both of these plants tend to emerge at around the same time. So not only are they active in the same places, at least in the Northeast, they tend to be really active at the same time. What you'll notice, and I'm not going to talk about this too much, but some of the work that we've been doing the past couple of years is trying to define understory plants by their phenological strategies. And Erythronium here is what we would consider to be more or less a true spring ephemeral. It really is only active for a period of a few months before it retreats back into dormancy, back into these below ground structures. So corms and bulbs and stuff, where it will then wait out for the next year. Whereas jack-in-the-pulpit has more of a summer green strategy where it still makes use of this highlight availability in early spring, but then it will continue to flower and fruit and conduct a lot more of its life history over the rest of the year as well. So it has a much longer activity period. But you'd imagine that in spring, there might be a lot more competition between these two species if they do occur in the same place than later in the year, where there's going to be less competition for resources after Erythronium retreats back into dormancy. And so again, this phenology component, this are they present at the same time, and how does this affect their biotic interactions? How does this affect how they interact through space and time is the main focus of my talk today. And so I keep using a term, phenology. So just to really nail down what I mean by this, phenology is a term that refers to the timing of repeated life history events. So anything that happens on a repeated cycle through time that is important to the demography and the ecology of an organism. It doesn't have to be the same exact time, but relatively on a cyclical process. We term that as a phenological event. And so for plants specifically, we tend to think about life history events like fruiting, leaf out, senescence, and flowering, things that happen roughly the same time every single year and repeated throughout the lifespan of those plants. Most plant phenophases, which is just a term that we use to describe these events, happen around the same time each year with variation. and the timing is determined by a combination of genetics as well as phenotypic plasticity. So don't have a whole lot of time to talk about this today, but just it's important to note that there are adaptive constraints as well as a certain amount of variation where these plants can respond without adaptation to a variety, a range of environmental conditions. So if any of you have a tree that you watch outside your kitchen window, for example, you've probably noticed that the timing of flowering varies year to year. That's not because that individual tree is changing its genetics year to year. It's because built into its genetics, its behavior has a certain amount of plasticity where it can leaf out earlier in warmer years, it can flower earlier in warmer years. It doesn't need to adapt to do so. And specifically what I'm going to be talking about today and where a lot of my interest really falls is in spring phenology. Again, this is something that I got really interested in, just seeing how much spring light availability and snow melt and pulses of precipitation, especially during this period of time, really seemed to strongly determine what plants I saw at a given time, where I saw them, and how they interacted with each other. And importantly, spring phenology, and especially leaf out and flowering, tend to be really strongly cued by both temperature and in certain systems, especially in the Intermountain West, snowmelt. Today I'm going to be focusing primarily on temperature. Events tend to be earlier, both with warmer springs, and then there's also a certain amount of evidence that cold winters also tend to shift phenology earlier in the year. So not only do warmer springs sort of tell a tree or a plant that we're kind of beyond the threshold where you really need to worry about frost risk, but cold winters tend to also cue that it's safe for the plants to become active in spring. We're not exactly sure how that works mechanistically, but does shake out in a lot of our statistical models. And so within this framework, a lot of what I'm interested in is how climate change will affect these phenological events, these phenophases. And I don't have to explain this to anybody here probably, but we know that environmental conditions are changing and that conditions are warming generally, especially in the northern hemisphere. We know that spring especially tends to have they tend to be a lot warmer now than they have historically. And we have been able to at least statistically link, but also mechanistically, that this change in temperature affects species phenology. And we can do this, at least statistically, going back hundreds, if not thousands of years in some cases, with continuous data sets like the Kyoto cherry tree blossoming data set that's shown here. And so this is year on the bottom. So we're here in, well, it cuts off at 2021, but the data set goes back to about the year 800, where people record the first day that cherry blossoms bloom in Kyoto. And you can see it's relatively constant, or at least a fair amount of variation around a pretty flat trend for most of this, before it's been moving a lot earlier in the year. We see this negative trend where lower on the y-axis here corresponds to earlier in the growing season. And that has really taken off around the same time that we see this sharp increase in temperatures globally and locally. And there have been a lot of synthesis papers. These phenological shifts have been really widely documented across a lot of different systems throughout the world, and changes in the timing of activity correspond really strongly to changes in environmental conditions. This is one of my favorite examples of this from a paper that was published in 2019 by Piao that found that at least looking at the northern hemisphere, you can see here using remote sense data sets, that in about 75% of cases, spring phenology has moved earlier in the year. And it's not exclusively that, but this is a very common trend that we tend to see worldwide. To make things a little bit more complicated, not only do we see these earlier shifts, but we also see species-specific sensitivities to environmental conditions. So species tend to react differently to they can either respond to a different set of environmental cues, or they can respond to the same cues in different ways. And so this is true both within functional groups. So if we're considering wildflowers as a group, Different spring wildflowers have different phenological sensitivities, but this is also true across trophic levels too. So if we think about how the sensitivity of a flower compares to the sensitivity of a bee or of deer, those tend to be different as well. And so this is an example of from a paper that was just published a few years ago in Journal of Ecology. And you can see here that the dark green here are trees, the diamonds. These squares are shrubs. Wildflowers here are in light green. So we see a lot of variation by, and each point here represents a different species. So, and on the y-axis here is the phenological sensitivity. So the more negative this number, the earlier they're moving per degree Celsius of temperature change. And so we see a lot of variation both between groups, so for example, between wildflowers and trees, but also within each group. So within these diamonds, for example, we see a lot of variation in how each species is responding to climate change, or responding to spring temperature at least. This is another great example from a paper published by Heather Karuba in 2015. And what she found was that wildflower phenology and butterfly phenology are becoming mismatched in time. Whereas if they were responding at the same rate to warming, we would expect all of these points here on the right to follow along this one-to-one line, but they don't. What this suggests instead is that plants are tracking variation in environmental conditions, but butterflies are largely not. And this could suggest that these flowers will become active early in spring, and the butterflies may not be there to make use of that resource and to help pollinate these flowers. And this type of dynamic where the biotic interaction becomes offset in time, we tend to refer to as phenological mismatch. So this is when species either that have previously interacted shift in phenology so that they no longer interact as much, or it can also be when species that historically have not interacted much shift their phenology such that they do interact a lot more. And so both of those can be problematic for different reasons. An example of this is if we have two facilitative species that are adapted to be active at approximately the same time every year. So for example, plants and pollinators, where the pollinators really benefit from a nectar resource, the plants really benefit from being pollinated. If we have, for example, trillium here shown by this blue distribution that generally represents activity. So starts around this time of year, ends a little bit later in the year, and then peak flowering would correspond to the peak of this curve. And then historically, what we would expect to see, or what we have seen, is that pollinators that interact really strongly with the species tend to be active at about the same amount of time. The duration of this interaction lasts for basically the duration that both of these species are active, and the peaks in activity tend to overlap really strongly as well. And so we can also not only get information about their phenology, but also quantify the amount that they overlap in time. And we know from extensive studies, again, that these two periods are moving early in time. So flowering is happening earlier in the year. Bee emergence is also happening earlier in the year. But it becomes a question of, are they moving earlier at the same rate, or do they have the same responsiveness to shared environmental cues? And if they don't, it might look something like this, where wildflowers are shifting a lot earlier in the year. And bees, although they are shifting earlier, they're not shifting as early in the year. And instead, if we were to quantify the duration of overlap, it would be much shorter. So even though their duration of activity above ground has not changed, the width of these distributions has not changed, the amount that those distributions overlap have changed and would signal that they are maybe not interacting as strongly over time. And so today, I'm going to be talking about two types of phenological mismatch. The first one talking specifically about phenological escape and the relation of understory plants to spring light availability and the timing of canopy close. And then I'm going to follow up by talking about some of the research that I'm trying to get started at my new lab in Tennessee, which is looking at dioecious plants, which are plants that have separate male and female individuals, and whether the phenology of their flowering is becoming mismatched in time as well. So phenological mismatch serve two ways. And so to begin with, just jump straight into it, one of the things that I've been really keen to explore for the better part of the past decade now, which doesn't feel like it's been that long, so it's a little weird to say. But this is work that I started getting really interested in during my PhD and have really sort of built out a little bit since then. And the basic gist of this section is that, as I mentioned earlier, understory plants tend to emerge up to several weeks before the canopy closes. They rely on this access to high spring light availability to fund their annual performance. So they really go all out in maximizing their photosynthetic rates and gaining a lot of carbon that they can then use to fund their metabolism throughout the rest of the year, even under dark, shady conditions. And one of the main questions that I'm interested in with this research is whether or not spring light availability is changing because of differences in phenological sensitivity between understory plants and canopy trees, and how does this affect the availability of this really important resource, and how does this affect the demography of the understory plants. And so just to go into this in a little bit more detail, understory plants emerge from dormancy up to several weeks. This should probably say up to more than a month before the canopy closes in some cases. So we're probably still a month or two off up here, but I can tell you that I'm getting very nervous about the beginning of my field season down in Tennessee. I was expecting to have until mid-March or so, but I was digging a soil pit last week and found some trout lily starting to poke out of their corms. So I'm very nervous and need to get out there and start recording data. But as you can see in this picture, this is trout lily here in the foreground, and you can see that it is fully flowering. This was probably a week or two after I noticed that it was starting to become active in the understory. And you can see in the background all of this blue sky that is still not being shaded by the canopy. And so they are, in this case, this picture was taken in Southeast Michigan. They will probably not be shaded here for another month or two. So they have a really long period of time where they have access to a lot of light and can be really photosynthetically active. This period of highlight availability is combined with really upregulated photosynthetic rates. So they really do go all out in their photosynthetic machinery to try to maximize the amount of carbon that they're assimilating per unit time, especially for these species that then retreat back into dormancy once the shade starts. So unlike tree seedlings, which are active throughout the rest of the year, this species strategy is just to be active while the light's there. And then why keep above ground structures if you're not doing anything with it? So they retreat it back below ground. And this graph here on the right is from a paper that was published in 2019 by Mason Heberlein. And what it shows is the rate of carbon accumulation throughout the year for a group of spring active wildflowers. And so all of these lines begin at 0% and go to 100%. So they're all scaled differently depending on the total carbon that's assimilated by each of these species throughout time. And you can see that so they start at 0, they accumulate some carbon throughout the year, and whenever they senesce again, so lose their leaves or just retreat back below ground, that's where the line ends. And so the difference on the x-axis between the start and the end is going to be the duration that they are active above ground. And then the slope of the line is, of course, the efficiency with which they're accumulating carbon. So the steeper the line, the more carbon they're accumulating per unit time, scaled within species, of course. But what is really important to see with this chart is that pretty much all of these species, and all of them are active in early spring before the shady period that's represented with this dark green box, all of these species accumulate at least about 50% of their annual carbon budget within a period of time that for some of these species is only 2 weeks long, 2 or 3 weeks long, but they're still getting more than half of their carbon in this really short period of time, and that's got to help last them throughout the rest of the year. Other species like Claytonia here is one of those true spring ephemerals that retreats back into dormancy before the light disappears. It maximizes, you can see this line's really straight. It does not waver It does not change its photosynthetic apparatus at all throughout this early spring period. It is just going all out to assimilate as much carbon as possible in this really short period of time. Other species like Trillium, which is this blue line, it persists a little bit into summer to fruit, to flower and then fruit. And you can see that through a combination of downregulating the photosynthetic mechanisms, but also just in a lack of resource availabilities, this line flattens out a little bit. It is not as efficient at assimilating carbon. And so it lasts later, but it's not doing as much with its time below ground. And then even other species like Hydrophyllum here in yellow flattens out completely into late summer, but it persists late enough into fall so that when the canopy reopens as the canopy trees senesce their leaves, we see it pick up back again and gain some carbon later in the year as well. All of this to say, though, is that all of these plants that are spring active, and this is a really common strategy, really rely on this period of high light availability in spring to assimilate the majority of their annual carbon budgets. And for spring ephemerals, this tends to be between 50, or spring active wildflowers, this tends to be between 50 to 100% of their annual carbon is within this period of time. But even for woody plants, so for some of the tree seedlings that I'm going to be talking about in a few slides, the value is 60 to 80% of their annual carbon budget is assimilated within about 2.5 weeks before the canopy closes. So it's like going home for the holidays, eating your weight in food, and then just being able to persist on that for the rest of the year, maybe a couple snacks in between. But for the most part, you just got to max out your consumption and then be able to persist on those calories throughout the year. And that's the strategy that these plants are employing. And this is really important because we know in several instances that carbon is the unit by which plants live, right? So they use, and for pretty much all organisms, we use carbon to fund our metabolism. We use it to construct new tissue. We use it to allocate towards reproduction and to have healthier, larger offspring. And so what I have found in a lot of my research, just to sort of summarize this and not take too much time, is that earlier emergence, so the earlier that these plants emerge in spring, tends to mean increased access to light. Increased access to light means greater carbon assimilation. So this is from a study I did as part of my PhD. Acer saccharum is sugar maple, Quercus rubra is northern red oak. What these lines show is that the earlier, so the further left on the x-axis, the earlier they leafed out, the more carbon they were able to assimilate on an annual basis, but especially in spring. And with greater carbon assimilation, so the higher this number is on the y-axis, the higher, the more growth that they were able to accomplish on an annual basis, and also the more likely they were to survive on an annual basis. And so these are survival curves for the same two species. The more carbon they were able to assimilate, and this is annual, but again, most of this is coming from spring, the more likely they were able to survive. And so this has strong implications for demography, population growth, tree recruitment, et cetera, all of these things that we're interested in from a forest ecological perspective. And I would argue as well, and there have been a couple papers that have come out recently making the case for this, that this is such a strong, has such strong importance for spring active plants in general that it really shapes not only their behavior, but also potentially their distributions. And this is one of my favorite graphs to show this. This was published by Augsburger and Salk in 2017. And what they were interested in was quantifying how frost and shade interact to limit spring wildflower activity periods. And so we see these 6 horizontal lines in the middle. So those represent these 6 different species of spring wildflowers at a site in Illinois. This dashed black line represents frost risk. It starts really high in winter, of course, and then as we move closer to spring and summer, the likelihood of these plants encountering a frost approaches 0. And then this solid black line represents the percent shade. And you can see it does not start at 0 because we still have those branches and trunks in the canopy. But as these trees leaf out, it gets shadier and shadier until it reaches 100%. And what you'll notice is that in between these two constraints are the activity periods of all of these wildflowers. Some of them may start a little early, maybe they're a little bit more cold hardy. Some of them might go a little bit later than this. Allium, for example, it will drop all its leaves, but it is just a stalk that has fruit on it. So it's not really photosynthetically active, but it's a little bit hard to quantify that just by saying if it's above ground, it's active. Anyway, all of this to say, though, is that we have this golden window of opportunity for these wildflower species, and it really shapes their biology, and I would also argue their biogeography. And so all of this sort of leads to the question, will climate change alter phenological escape? So phenological escape being the strategy of avoiding shade by being active really early in the spring. And so the question becomes, will canopy trees have a stronger sensitivity or a weaker sensitivity to the same environmental cues as these understory plants? And so earlier when I showed these distributions, I gave an example of plants and pollinators that are moving apart in time, but the question here is almost the opposite. Are they moving together in time to interact more strongly? And so we know that both of these groups are moving earlier in the year with respect to their activity, but are they moving forward at the same time? And if they're not, if, for example, trees are much more sensitive than wildflowers, what we'll see is a lot stronger overlap in the timing of their activity and the duration of that overlap period, which would indicate that these understory plants are being shaded for a larger portion of the year. And as we've established over the past couple slides, that's not great. They need that period of open light access to really fund their metabolism. And if they can't do that, it has negative implications for the demography and for their fitness. And so what do we find? Just to jump straight into results. It depends a lot. I mentioned earlier all of these differences in species level sensitivities, differences in sensitivity between functional groups. So it really depends on which species you're interested in, and getting these measurements on a species or other sort of phylogenetic information about the sensitivity. This is another figure from my dissertation. The blue and yellow lines are the tree seedling phenological responses. So on the y-axis, we have the day of the year. And so as these lines decrease or move closer to the bottom, it indicates that the relative timing of their phenology is moving earlier in the year. And we have average spring temperature here on the x-axis. So all three lines, including this canopy line in green, are moving earlier in the year. And it's a little hard to tell from this figure. I apologize for that. But the distance between these lines is increasing, indicating that tree seedlings, at least in this study, were found to be more sensitive to warming than canopy trees, which is good news if you're a tree seedling. It means you're more likely to have increased access to spring light over time, assuming warmer temperatures. So that is a good thing from the perspective of an understory plant. However, if you conduct a similar study with spring wildflowers, for example, and this is another one of Mason Heberling's papers from 2019. Here, the solid black line is trees, and the dashed gray line is wildflower sensitivity. And so you can see, again, they're both moving earlier in the year in response to warming temperatures, but those lines are converging, suggesting that those periods of activity are moving closer together and that wildflowers will not have access to as much light over time, assuming these warmer temperatures continue into the future, which I would argue is a pretty safe bet at this point. It also matters about, or it also seems to depend on biogeography as well. So this is a paper that we published a few years ago looking at these patterns for spring wildflowers across different continents. And what we see in North America is largely the same story that Mason was showing in 2019, which is that anything in these cool blue colors indicates a region in which the period of highlight availability is expected to decrease relative to wildflower activity periods. So these are areas where we would expect to see more shading and potentially lower wildflower performance in response to warming spring temperatures. Contrast that to Europe, which is largely these neutral sort of yellow, orangey, maybe slightly positive values. So wildflowers might gain a little bit of access to spring light, or in Asia where we see these really strong darker red, orange colors indicating stronger gains in access to light. it seems that there might be differences in evolutionary constraints or adaptation to different amounts of variation to seasonal environmental conditions that may have set up differences in what we would expect for phenological escape across these three continents. Another really important thing that I just want to touch on here is that it really depends on the data. So although here we were showing decreases, at least in North America, to access to light, there have been a couple recent studies. And importantly, this study was done using herbarium specimens, specifically, at least from the understory, well, no, for the understory and the canopy tree perspective. This is a paper that we're preparing right now that's being led by Clayton Hale, who's down at the University of Georgia. And what he found, or what he is finding currently, is that wildflowers are actually more sensitive. The slopes of these lines, I'll just point out the scales are different, so it doesn't look that way just eyeballing it. But the wildflowers are shifting earlier at a rate of about 3.6 days per degree Celsius, whereas the trees are only shifting forward at a rate of about 2.8 degrees, or 2.8 days per degree Celsius. And what he is using is he's using community science data for the wildflowers, and he's using remote sensed MODIS data for the canopy close. And so we see the opposite trend projected from these data, where wildflowers would potentially increase their access to spring light under warming conditions. And one of the things that we don't know is why we get these different signals depending on the different data sets that we look at. In fact, this figure comes from a paper that I published last year, where we specifically asked that question. There were two papers that were published in Journal of Ecology, basically a month apart from each other in 2023. One of them was using herbarium data. The other one was using community science data from the National Phenology Network. Almost identical questions, almost identical models, but they found opposite signals. And I really wanted to know why. And so this was actually a really cool collaborative project where I reached out to both of the groups, and they were very gracious in letting me pick apart their data sets and then see if I could reconcile the differences just statistically and potentially just cropping the data in different ways so that they matched in space and time and all of that. And it was something that we were not fully able to resolve is the short version of that answer. But you can see for community science, we see that these lines are diverging over time, indicating that wildflowers should potentially increase access to light as temperatures warm, whereas the herbarium signal, even after making the models identical and cropping the data sets in different ways, showed either no change, these lines are basically parallel, or maybe even slightly converging, which would indicate a reduction in period of light availability. But so opposite signals, and it seems to really depend on the type of data, and we don't fully understand that, and it's something that we should keep in mind when interpreting phenological data, is that the way that data is accumulated seems to matter. And a quick plug, if you're interested in this topic, I will be giving another talk in about a month from now with the National Phenological Network. There will be a webinar March 21st. We just figured out this morning what day it's actually going to be. So I don't have a link for you all yet, but if you're interested in learning more about this aspect of the research, I'd encourage you to look it up. And so with all of this change, it depends on the species, it depends on the data and everything. Will it actually matter to demography? The short answer is yes. I'm seeing you standing up in the back of the room. I'll hustle my way through this. For the tree seedlings, we see that even if they do move earlier in the year, this black line represents current conditions. This red line is extreme climate change conditions, and moderate climate change in orange. So when we project this into the future, we see earlier beginnings, but we see that the physiology, the photosynthetic behavior really varies. Or even if they're shifting earlier, if they're less efficient, or if there's extreme warming and respiration demands, that can affect their fitness as well. And climate change will severely stress tree seedlings, even with these gains in light availability. Wildflowers may not be so lucky. Do I have time to go into like 5 or 10 minutes? Yeah, okay. So just really quickly, I want to plug the second phenological mismatch, which we've been working on for the past few years and hoping to build up over the next 5 to 10 as well, which is looking at dioecious reproduction. I'm going to cruise through this. So dioecious plants have male and female flowers on separate individuals. And recent papers have suggested that male and female plants are differently sensitive to warming. So specifically, male plants tend to be more sensitive than female plants. So we might see a mismatch where they're no longer overlapping in time as much, which has negative implications for reproduction. And there's also this third component, which is that they're, at least the species I'm interested in, which is spicebush, is animal pollinated. So we also have to worry about how the bees fit into this as well. And what we're interested in is how it affects reproduction and how it affects demography. This is a common strategy that implicates obligate outcrossing. So evolutionary is kind of interesting as well. And I'm just going to plug this right here, and it's something that we really don't know how this plays into it. But there is evidence that dioecious plants can switch sexes under certain conditions, and we're not really sure if that's environmentally correlated or not yet for certain species. But I'm assuming most of you have seen ginkgos before, pretty common street tree, pretty ancient genus. It's dioecious. It is widely documented to switch sexes. And we know this because the fruits are disgusting. All right, they smell like dog poop. They're awful. And so what urban planners have tried to do for a while now is only to plant male trees. And that's what they've been doing. But if they're only planting male trees, why do we see fruit along these streets? And it's because they will switch sexes, and it seems to be linked to environmental stress, so particularly dry and warm conditions. So this also complicates matters of how do we assess phenology if under certain conditions, maybe they just switch sex instead of continuing that earlier trend. We know that these male and female plants are separate in time. Male plants tend to flower earlier, but they overlap for several weeks where flowers are active for both sexes. And we need, in order to have successful reproduction, they do need to match up for some period of time along with their obligate pollinators. This looks like mismatch is happening. This is a really cool paper. I won't get into it too much, but please look it up. Just came out last year. And so what I'm really interested in trying to understand better is how this mismatch, if it's occurring, will affect the fitness of the spicebush that we see up and down the East Coast. From a preliminary study, we know that they are sensitive to environmental change, but we want to really test this rigorously with field data and not just rely on herbarium specimens, for example. We also, so we're setting up an observational network across the eastern US. We've been tracking individuals and population level metrics for the past 2 years or so. We're going to continue that into the future. And one of the main things that I'm really interested in doing is measuring and collecting fruit to get at this reproductive component. We'll also be instituting an experimental treatment where we are bagging female flowers for different periods of time in early spring to approximate differences in phenological overlap. So if we leave the flower on for X number of weeks, that would simulate shorter accessibility to male pollen, for example, and see if that affects reproductive output as well. And so more to come. That's going to be coming over the next 5 to 10 years, but I encourage you to keep an eye out for it. And just some quick take-home messages. Why should you care? Spring wildflowers are really highly diverse. Herbaceous plants constitute about 80% of the botanical diversity in temperate forests. And spring ephemeral wildflowers make up about 20% of that diversity. So about 16% of total herbaceous diversity or botanical diversity in northern temperate forests are these spring ephemerals specifically. And that's not even including species like Trillium that maintain their flowers a little bit into the summer. This is true spring ephemerals that are only active for a few weeks or maybe a month out of the growing season. And it really strongly shapes their biogeography. This is another paper that we published last year led by Abby Yancey. And so we see, for example, really high concentrations of spring ephemerals in southern Appalachia especially. So this will have local implications as well, or regional implications as well. When we think about pollinator resources, if plants and pollinators become mismatched in time, this will affect not only the plant reproductive success, but also the animal ability to obtain the energy it needs to conduct its metabolism. And it also can be mismatched just in time. Again, a lot of these bees really rely on highlight availability to be able to navigate these forests, and they tend to avoid really shady conditions. So even if they are active and there's too much shade, they might just not be pollinating these forest plants. And lastly, because I care a lot about trees, and I know a lot of you care about trees, the ways in which tree seedlings are affected by climate change and by this phenological escape success will determine what future forests look like as well. So when we think about recruitment processes where we start with seeds, they recruit to some cohort of seedlings, to saplings, to juvenile trees, and finally to adults, this pathway that we get to from the seeds being present in the sand to what actually composes the forest canopy and then feeds back to affect all of these interactions is really strongly determined, especially at the seedling stage, which is where we see the vast majority of directional change in species composition. So what future forests look like will be strongly implicated by what happens now for the tree seedlings of today. With that, a lot of acknowledgements. All of these people really helped in some capacity to all of the work that I talked about today. NSF funded a lot of this research. Many thanks to the various programs that have been hosted me as I do this research, especially these postdoc institutions, which is where I've been for the past several years, 3 or 4 years. These are where all of the data came from today, various sources. GBIF really represents all of the museums. I can't fit all of the museums that I pulled herbarium data from on a single page. So that's kind of the stand-in for there. And my new home, which is East Tennessee State University. If you're interested in learning more about my research, I'd also just like to plug my new lab is the Plant Ecology and Climate Change Lab, the Peach Lab at East Tennessee State University. Please feel free to send me an email if you have any questions that I don't get to today, or to just check out my website. Thank you for attending, and I'd be happy to take any questions. And this is Jack Hatayek, who's right here, who's a master's student at Yale, who I was lucky enough to be able to work with during my time at Pitt while he was an undergrad there. So.
I have a question from Zoom
Sure.
from Jesse. I was wondering if you have an idea to what extent photoperiodicity plays a role in climate change responses. Does it limit the plasticity in certain species, or does it help it buffer and keep interactions intact?
That's a really great question. So photoperiod is kind of this interesting phenological cue that it tends to be one of the only cues that is more or less going to be the same every single year. So because it's shaped by the rotation of the Earth and the latitude that you're at, for a given plant, photoperiod is going to be the same on a specific day of the year, pending cloudiness and all of that. But it's not going to vary much as a signal. That being said, it seems to be a very strong signal when we look at latitudinal differences. So as we move north to the north and photoperiod shrinks on any given day of the year, we tend to see more conservative phenology where plants become active later in the growing season. And that's also a measure of temperature as well. Of course, northern latitudes tend to have colder temperatures, but even when we do like growth chamber experiments to try to parse those apart, we do see that photoperiod does play a really strong role. Now, one of the main roles that it plays in cueing phenology is if we have, let's say, a very warm winter and a very cool spring, where the plants don't have the same like accumulation of degree days to trigger their phenology, which is kind of mechanistically how the warming temperatures trigger phenology. The photoperiod, there seems to be some sort of like, if nothing else, we need to go by this day. And so that seems to be the real photoperiodic mechanism that plays into that. And so at the end of the day, if nothing else triggers the activity of these species, photoperiod will, which can help keep those interactions going, even in a really bizarre, at least environmentally bizarre year. So the answer to the second part of the question, like what does it mean for the interactions, is I think it, at least on an evolutionary scale and for adaptive reasons, it does really contribute to keeping these biotic interactions together.
We have time for 1 or 2 more. Thanks for the talk, Ben. Could you go over a little bit more on the use of herbarium specimens and how you can use that for these types of studies?
Sure, yeah. So herbarium specimens, this is one shown here for Trillium erectum, I believe. So red Trillium. These are really valuable to phenological studies. And so one of my postdocs was working at the Carnegie Museum of Natural History in Pittsburgh, which these are great for phenology because you can see preserved in perpetuity here that this plant was flowering. And what you'll also notice, if you can read really fine blurry text, is this says April 20th, 1980 was when this plant was collected. And so we have the ability now to estimate environmental conditions if we have a date and a location. The location is described here. It tends to be pretty narrowly defined when we use it for this type of research. So if we have a location, we have a date that it was collected, we know that it was flowering on this date. We can then estimate the environmental conditions during that year and use it to model change in the timing of phenology over time. And there's a lot of research within the past 15 or 20 years that have really used these herbarium specimens and shown that they are very accurate to trends that we can measure in the field as well. So this it's extremely useful for creating longer term trends and having an idea of like how environmental variation affects the timing of activity when otherwise we might have to conduct like a 10-year study to really be sure and have enough data to put that all together. So they're really cool. Working in an herbarium is awesome. It's like a plant library. So it's very relaxing. Light tends not to be great, but it's a nice quiet place to just kind of hang out and work with plants. So I encourage you to all check it out if you haven't already. Cool. Thank you all for attending.